Hello, my beautiful and intellectually curious and compassionate love bugs. Today, I implore you for a little bit of extra compassion as we talk about the Gabby Petito case. Please remember that Gabby Petito and Brian Laundrie are people, and they have families and loved ones who are also people that are going through an incredibly difficult time just with the news, processing everything that's happening, grieving, and also dealing with the intense media presence and what is being said about them, their families, and their family members. This case really struck a chord with me because my advisor, who even suggested that I should teach the forensic entomology class, more on that in a second, or that I wanted to, she was murdered in a domestic violence incident, and it was caught in the media in the area that I was working and studying, and many things that the media were saying were so disrespectful to her and to her family and those of us that were going through the grieving process. It really just only made it worse. We are going to be talking about this case as respectfully as possible and keeping all the people involved in our hearts. I have seen a lot of YouTubers kind of muck up talking about this situation, so we are going to do it as respectfully as we can and just talk about the science. I wanted to do this because I've seen a bunch of questions about the Gabby Petito case. Specifically, why is Gabby's time of death somewhere between August 27th and August 29th? Why don't we really know? And when people are talking about the remains of Brian Laundrie that were found, people are questioning how the body could be in a skeletal state in just 4 weeks. Especially if you just Google online and you're like, Google says it takes a lot longer for a body to decompose, so obviously some conspiracies are happening. So today I wanted to talk about what forensic entomology is, what it isn't, how it works, and a little bit of its limitations to help answer those questions so you can see what is within the realm of possibilities. What makes me qualified to talk about this? I'm glad you asked. Hi, my name is Nancy, if you haven't been here before. Welcome back to my channel. I am an entomologist, which means that I study insects, and I live in Ecuador, where normally I'm doing bug tours, but sometimes I'm on YouTube talking about insect biology. I have my master's degree in entomology from the University of Georgia, and I taught forensic entomology as the instructor of record for 2 semesters at the University of Georgia. I taught fall 2012 and I taught fall 2013, and it garnered enough attention that my Entomological Society of America wrote an article about it. I'm not saying that I'm an expert that has like 20 years in the field, but I definitely have enough knowledge to answer some of these common questions. That is enough bragging about me. To give you some better examples and ideas about what is possible in different time frames, I will be using pictures from student experiments during that class. I do have permission to use all these photos. During that forensic entomology class, students could put pigs into the field and see how they decomposed after 5 weeks in different varying circumstances, like if the pig was buried, or if the pig was broken up into pieces, or the pig was cut open. And I will be showing pictures from that experiment. I will tell you verbally and I will tell you on screen that that is coming, so that way if that's not something you want to see, you can just like look away, turn to another video, just listen, you know, whatever works for you. But you will see animals in high level stages of decomposition. That's the reality of this science. Whew, that was a lot to get through, so let's start. First off, what is forensic entomology? Forensic entomology is a branch of forensic science which applies to the study of arthropods and insects in the investigation of civil and criminal matters. So, as I just mentioned, there are 2 branches of forensic entomology. Civil is like, there are termites in my apartment, or there's a worm in my sandwich, who pays for this? And criminal is, I found a dead body, that one's been murdered. How do we determine how long they've been out there? This is bones, CSI, kind of movie stuff. So we are obviously going to be focusing on the criminal aspect versus the civil. If the body is found within 72 hours, it can usually be pinpointed to at least a pretty general time of death. That is because we know how bodies systematically cool after a certain period of time, and we know when rigor mortis sets in. That is not in the realm of forensic entomology, so I'm not going to comment any further on that because I don't want to be an idiot. The goal of a forensic entomologist is to determine something called the PMI. The PMI is the time elapsed since death, and forensic entomologists are looking for the minimum PMI or the minimum time elapsed since death. And this usually is calculated within a range. That range can be estimated to a few days to maybe a week, or sometimes, depending if there's not a lot of evidence or not, even whole months or maybe even a season. The more evidence you have, the sooner you find the body, the more specific your postmortem interval is going to be. A PMI can never give an exact time of death because it uses biological factors, ecological factors, and abiotic factors such as weather, which are unpredictable. And so you can do the best you can to have all the relevant information, but there is variation in all of those things. Forensic entomology is basically applied ecology. If we look at the body as basically a mini ecosystem, it goes through several different stages of what we call succession. Just like a forest would go through succession, bodies too will have different states, different factors, and different animals and insects that are colonizing it. In the beginning, we have the fresh stage. The body has just been put out. This is when flies start to come lay eggs on the body and when bacteria starts to break it down. Then we have the bloat stage, which when the gases from the bacteria build up sufficiently that it causes the body to swell. Usually in this case, it's pretty easy to identify who or what this thing was. Then we get into advanced decay, where it can be a little bit more difficult to identify the person because now you're getting down, the body's being broken down into fluids, and there's not a whole lot that may be recognizable of that person. During post-decay, you have hair, you have bone, you have tendons, you have more of those tissues that are more difficult to break down. If you do have any soft tissue material left, it's most likely turned into a hard, waxy substance at this point. Skeletal stage, which is just the skeleton, as the name might suggest. During each of these different periods, you will find different insects at different life stages. The earlier that you find the body, the easier it is to calculate the PMI, and the shorter that interval of potential death is. The PMI is determined by 5 major factors. The first is the species of insects you find and the life stage they're at. Flies tend to colonize the body first, and beetles tend to colonize later. Most of our data comes from the development of flies, and so if you get past a certain stage where there's just beetles, we don't have a lot of data from, it's harder to calculate the PMI, and is way more difficult to calculate the PMI after you get to the skeletal stage where all the insects have left. You need to know the stage of the insect that you found it at. Did you find it at a larval stage? Did you find it at a pupal stage? What species did you find? Because, for example, some flies give birth to live young and some flies give birth to eggs. You need to know what the lifestyle is and what life stage you found it in. You need to know the temperature at the crime scene, and you need to know the weather station data for that area. You may be wondering why we need to even measure the temperature at the crime scene if we have weather station data. This is because your crime scene may have a different temperature than the ambient temperature from the weather station data. For example, if you find your body in the back of a car, or you find your body inside, or it's covered under debris or blankets, that temperature will be different than the ambient temperature that the weather station recorded. And the 5th thing you need to know is the growth rates of the insects. You need to know how long it takes for the insect to go from the egg stage to the first larval stage, from the first larval stage to the second, etc., at different temperatures. Insects are cold-blooded, which means that they regulate their body temperature based on the environment around them. So the higher the temperature, the faster their metabolism and the quicker they develop. Let's go over the steps that you need to take to determine your postmortem interval. The first thing you need to do is collect the insect evidence and be very sure that you have checked everywhere, including under the body and the surrounding area. Many insects will feed during the larval stage, but then move to pupate. So if you miss the pupal casings, you're going to get a PMI that's too early. If you have insects that are only in the larval stage, you will need to take some of them and rear them out so you can identify them. Fly larvae cannot be identified as larvae. You do need the adult to do the identification. The second thing that you're going to do is you're going to take a data logger, which will record ambient temperature every 15 minutes and put it at the crime scene for the next week. This means if you have a car, you're putting it in the trunk of that car to see how the trunk of the car heats up differently and cools differently than the ambient temperature. The third thing you do is you get weather station data. You compare the weather station data from the week that you put your data logger at, because you can directly see if the ambient temperature is X, the crime scene temperature is Y. And you can create a correction factor that allows you to use that formula to back calculate what the temperature at the crime scene was during the time that your person was missing. This is really important because the higher the temperature, the faster the insects develop. So you want a good correction factor so you can be as accurate as possible when determining what temperatures these insects were exposed to. The next thing that you are going to do is that you are going to research your insects. Each insect develops at a different rate at different temperatures. It's not generalized. So you are going to research the species that you found, and you are going to see how they develop in the lab when exposed to different temperatures. This data is collected and recorded and done in a unit called accumulated degree hours or accumulated degree days. You're basically asking the question, if my insect is exposed to temperature X, how many hours does it take for it to develop? And then you will get a number. You are then going to take your data sheet that has the correction factor so you know what the temperature the insects were experiencing during that time. And for each species, calculate the accumulated degree hours that they experience. Say it takes 4,000 hours for species X to reach its third larval instar. You'll basically find that in your sheet and be like, this is the earliest it could have developed, and this is the latest it could have developed. You do this for each of the species, for each of the life stages that you found it at. On screen, I just showed you the minimum time required that it would take for those insects to develop. But the PMI is always a range because those insects are not developing at max speed all the time. Temperatures fluctuate. They can fluctuate a lot, especially in like spring or fall where you get really, really warm afternoons and really cold evenings. Therefore, your range will be somewhere in the middle between the fastest those insects could develop and the slowest those insects could develop. So for example, you may have species A that developed until the third instar, but you may have species B that developed until the second instar, and you might have species C that developed until the pupal stage. But you know that species B colonizes way later than the other two. So using all of this information, you basically put together this big puzzle. This is why the longer the body is exposed, the less you have to work with. You have fewer insect species to work with, you have fewer specimens to work with, you may not have good quality specimens or specimens that we know a lot about their biology. Gabby Petito had a lot of things going for her. We knew the last time we saw her, we knew where the van was on certain dates, and we know when Brian had been seen without her. So even with just that, we had a pretty good window. Also, it seemed like her body was at least in a fairly decent stage. From the interview I watched with her parents, they say that they recognized her based on her clothing, but the body was still in good enough condition that the examiners could determine that she died by manual strangulation. So that suggests to me that her remains were intact enough to do a lot of this science. Her remains were sent to several different scientists who all work together, I'm sure, to put together the complete story, or as much of the story as possible, to give the family as many answers as possible. I know her body was definitely sent to the pathologist. I believe I read that they sent her remains to an anthropologist, and they did send her remains to a forensic entomologist. That, combined with the fact that we know when we last saw her and we know when we saw Brian without her. I mean, that's not saying that she wasn't alive at that point, but it's kind of suspicious, you know? So putting all of that together, plus the evidence from the forensic entomologist, who, by the way, gave a pretty short PMI. So her estimated time of death is somewhere between August 27th and August 29th. And as PMIs go, that's pretty specific, especially since her remains were found 3 weeks after the murder. Usually when you find a person 3 to 4 weeks after death, of course it depends on the state of decomposition, you're looking at a PMI of anywhere between 5 days and a week, and that's pretty normal. The longer the body is exposed, the more insect evidence there is, or the further along in decomposition the body is, the more difficult it is to calculate a PMI. And usually if you find a body about 4 weeks after the estimated time of death, you have a post-mortem interval of about a week. That's pretty normal. And in the Casey Anthony case, I don't know if anyone remembers that, a forensic entomologist was called in, and because there was so little insect evidence to go on, they could only really estimate her time of death to a month or even a season. So the more evidence you have, the quicker you find the body, the shorter that postmortem interval range is, but it's still a range. In the case of Brian Laundrie, partial skeletal remains were found. I know confirmed they found his skull, and that's how they identified him based on dental records, which again is not anything to do with forensic entomology, but the skeletal remains were very skeletal, and the area that they found him was previously underwater. That makes it very difficult because one, insects don't necessarily eat bones. There's not a lot of things that decompose bone. The other thing is if it was underwater, it must have washed away a lot of evidence, and now you have other things that may be contributing to the decomposition. Basically, forensic entomology cannot be done on skeletal remains. Aquatic insects, if you're like, oh, well, he was underwater, just use aquatic insects. Aquatic insects are not used in forensic entomology because they are generally not decomposers, and their biology is not known well enough, and we don't understand how bodies decompose well enough in the water to give a good answer. There are a lot of factors that could have affected or accelerated the decomposition of Brian Laundrie's remains. When we do forensic entomology experiments, we cage the pigs in because we don't want vultures or other animals tearing the body apart and making it more difficult to collect insect evidence. For those of you who are asking, we use pigs in forensic entomology experiments because they do not have fur like we do. They weigh about the same, so they decompose pretty similarly to people would. And that's why places like the Body Farm in Tennessee are so important to really help understand how bodies decompose under different circumstances, so that way we can solve cases better because we have better data. In the Florida swamps, you have vultures, gators, you have apparently wild pigs that someone said literally eat anything. So when he died, his body could have been torn apart, moved around, pieces moved significantly just by the initial animals that would have gone to the remains. I've seen partial skeletonization in just 2 weeks. In some circumstances, in extreme cases, a body can be skeletonized in just 9 days in really hot, humid, tropical environments. But for the most part, you can get down to a skeleton or just bones in anywhere between 4 and 5 weeks. There are several examples in where the body can take longer to decompose. This would be if there's lower insect diversity or lower insect activity. This could be at high altitudes or high latitudes during the winter or the fall where temperatures drop. Or this could even be if the body was covered or stored in something that insects didn't have access to. Florida has a lot going for it. It's hot and it's humid, and in that part of the reserve it's relatively undisturbed. So you have a lot of diversity and a lot of things that may be taking advantage of the situation. So there are a lot of factors that can affect decomposition, but it is definitely not outside the realm of possibilities that you could have skeletonization in a hot, humid area in 4 weeks. At this point moving forward, I am going to be talking about the student experiments in which we place the pigs out in the field, and there will be images of very decomposed pigs. They don't really look like pigs anymore. I don't know if that helps or not, but this is your trigger warning. As I was mentioning earlier, the student experiments, we put pigs out in the field for 5 weeks. The students could do whatever they wanted to their pigs. Some of them were very frustrated with the semester. The vet students decided to cut their pig open and see what would happen. We had 2 students who covered their pig with sticks and debris, and we had a couple students who broke their pigs up into itty-bitty pieces and kind of scattered them. The pigs were caged in, so if body parts would have moved due to insect evidence or animal evidence or just washed away by the rain, they were actually relatively confined because they were in a cage. The students who decided to break their pig up into pieces stopped at just the hind legs, and those had a much higher level of skeletonization than the rest of the body. And parts of their pig were skeletonized at between 3 and 4 weeks. What was interesting about the pig that the students had cut up was that there were pieces that were small enough that the maggots themselves or the insect evidence itself was moving the pieces. Again, they got stuck on the edge of the cage, but you could literally see the pieces moving because there were so many maggots moving it along. The students who left their pig relatively intact, but just cut down the middle of it, exposing the organs, their pig was practically just a skeleton and some skin after their 5 weeks. And the students who covered their pig had a high level of skeletonization. There was almost no tissue that was left besides bones. And I believe if that pig had not been caged in, you would have seen pieces of the skeleton get washed away because we did have heavy rains. So when we look at Brian's case, it's Florida, it rains, that part was underwater. They could have been pulled apart by various animals. I do not think it is outside the realm of possibility that they only found a few pieces of Brian Laundrie's remains and that those pieces were completely skeletonized. I hope that cleared up some questions or maybe some conspiracy theories that you may have seen on the internet about this. Remember that forensic entomology is a very educated assumption revolving around temperature and insect biology and general ecology, and there are cases where sometimes extreme things happen. Our data is very limited in some of these more extreme cases, and oftentimes if forensic entomologists are confronted or presented with difficult cases, they will reenact the crime scene with a dead pig to try and get as much information to be as close as possible to the potential crime scene situation, so that way they can give the best calculation they possibly can. This is why PMIs are never, unfortunately like the movies, are never a single day and a single time. It's because you really do have a range because you are dealing with the real world and ecology and biology, and it is complicated. Please remember to be kind, and if you are talking about the case, do it in a respectful manner, and I will see you all very soon in another video. If you liked this video and you want to see more forensic entomology stuff, kind of go a little bit more deep into various aspects, let me know. I love forensic entomology and I love talking about it, so let me know in the thought box. You can check out some more Entomologist Explains up there, and you can check out a video that the YouTube gods have decided that you would like down here. Don't forget to like and subscribe and do all of the things, and I will see you love bugs very soon in another video. Bye!